Hi. Hi, everyone. Hello. What an amazing day we just had. I feel like we had the entire season of summer in a day, weather-wise. It was wild. Anyway, welcome everybody to the Shoals Marine Laboratory weekly marine science seminar, otherwise known as the Rock Talk. I'm Jen Siva, your executive director, and just so for those of you in the room, so you know, this is happening online, this is happening in this room, and we are recording it. All of these talks go on to our website where you can see all the rock talkers of the past and future on there, which might overlap with some of your research projects or favorite topics. So I invite you to think about that and to think about when you leave here that you can come back here virtually through this format. So it's on our website, you can check it out. For those of you who are not aware of this, Shoals Marine Lab is the oldest and the largest undergraduate focused marine lab in the country. Yes. And it is jointly run by UNH. Come on, Wildcats. And Cornell. Do you want to do that over again, UNH? Every summer here at Shoals, we have these rock talks for our entire community, and we're really excited to share this one with you tonight. So our format tonight is 45 minutes of a fabulous talk about anemone, fish, clownfishes, and awesome behavior. And so 45 minutes of that, and then 15 minutes of Q&A. So save your questions, think of questions. Peter said he loves questions, so hand some to him. And if you have questions online, put them in the Q&A, and I will read them out loud to the whole group. And when we get to the Q&A, I'm going to bring you this nifty mic so you can feel like a rock star. So if you have any questions for the people online, you can put it in the chat and Casey will help you out there. I'm very, very happy to say that Peter Buston is here with us tonight. He is a professor in biology at Boston University. Yeah, thank you. He is also the director of BU's marine program, and his work largely focuses on behavior, evolution, and population biology in fish. I just feel like we've hit all the classes out here by that description alone. His lab asks questions at the frontier of behavior ecology, population biology, and evolution in the marine environment. He has an undergraduate degree in zoology from Oxford University, which you will detect in his accent when he gets up here. And he has a PhD in neurobiology and behavior from Cornell. Yeah, once again, there's a lot of big red love in this room right now. I also, my two little anecdotes, which he was shy about me saying, is that he's a half decent salsa dancer and a beginning meditator. So maybe you could ask him about that as well. All right, without further ado, here's Peter.
Thanks very much. Thanks all for having me. It's lovely to be out here. It's the first time that I've made it out here. Is this part of the tour? No, I can't tell you what's going on here right now. It's okay. Yeah, it's so lovely to be here. First time out here, despite doing my PhD at Cornell a long time ago. So thanks all for having me. So today I'm going to talk to you about social evolution in clownfish, and I'm happy to take questions as we go along, or you can save them to the end if you like. Okay, so first up, I want to talk about animal societies. And so animal societies are groups in which some individuals forego their own reproduction and help others to reproduce. And they're one of the more remarkable products of evolution. So these can be things like societies of ants or bees or wasps or termites, social cockroaches, snapping shrimp, naked mole rats, white-fronted bee eaters, cichlid fishes. They occur across the animal kingdom. And these societies were an evolutionary puzzle for Darwin. Right there in the middle of the origin of species, he calls them his one special difficulty, which at first appeared to me insuperable and actually fatal to my whole theory. And he was alluding to the neuters or sterile females in insect communities. And so to put this sort of problem in modern terms, problem is if individuals aren't reproducing, then they're not passing on their genes to the next generation, and those phenotypes should go extinct within one generation. So there's sort of two solutions proposed to this problem, and the first was proposed in the 1960s by W.D. Hamilton, and he had the insight that there are basically two ways for individuals to pass on their genes, right? So they could do it directly, by producing their own offspring, or indirectly by enhancing the offspring production of their relatives. So if, for instance, I were able to help my sister or brother have additional offspring, those offspring are not as related to me as my own offspring, but they are in some way related to me. So I can be putting my genes into the next generation in that way. We not only have to explain why, how individuals get their genes into the next generation, but why they don't go off and breed on their own like any other sort of self-respecting organism. And the insight there came from sort of Stephen Emlyn and others in the early 1980s. And he pointed out that there are two alternative options available to individuals. Either they can stay on their natal territory as non-breeders, helping their families or their relatives to reproduce, or they can disperse from their natal territory to breed elsewhere. And when those options for dispersal to breed elsewhere are poor, when there's ecological constraints, individuals might be more predisposed to stay home and help their relatives. This can all be summarized in an inequality that's called Hamilton's inequality. Both ideas can be captured in this inequality. And so if you look at this, the xi, you can think about being the non-breeder's fitness associated with its actions, the y terms being the breeder's fitness associated with the non-breeder's actions, and r being the probability of a sharing an allele identical by descent of the coefficient of relatedness. And so the I terms on the left-hand side of the inequality represent the payoff associated with the non-breeder staying. So the xi might be 0, it's not breeding, but it's getting some fitness payoff by helping its relatives to reproduce. And that payoff is discounted by relatedness. And on the right-hand side is the payoff associated with leaving. And so if there's some ecological constraint, such that those terms on the right are small, then the Ri, Yi can be greater than what's on the right. And so individuals can stay home and help their relatives to reproduce, even if they're not reproducing themselves. There's another way for individuals to get on their genes into the next generation. And that's directly by producing their own offspring in the future. And the idea here is if you go and you look at one of these animal societies at a particular moment in time, what you're seeing is a snapshot. So you might see individuals who aren't breeding, but if you come back, depending on the society, in months or years, you might find that those individuals have gone on to inherit the breeding positions in the future. And so that's another way for individuals to pass on their genes. There's also another option available to individuals, and that's not just, we mustn't just ask why they don't disperse to breed elsewhere, but we must also ask why they don't contest to breed at home on their natal territory. Why aren't they fighting with these other individuals to breed? And it's possible that in some cases they do fight and they win those fights and they get to breed and pass on their genes that way. So these ideas of sort of future selection and social constraints can be captured in a similar inequality. Individuals might stay home and forego their own reproduction. Xi might be 0. If there's some probability of breeding in the future, where F is the probability of breeding in the future, and if there's some social constraint such that the right-hand side of the inequality is close to 0, such that the probability of winning a fight is close to 0. Okay, that can be another reason why individuals will forego their own reproduction, wait at non-breeders, because they're going to inherit the territory in the future and they can't win a fight, so they tolerate it. So there's some limitations of this framework. Most of the theory was developed in parallel with ideas that were gleaned from working on terrestrial systems like birds and mammals and social insects. And so the generality at the time that I started my PhD was perhaps unclear. The number of taxa that had been studied was relatively limited. And so myself and other colleagues have set out to test the robustness of this sort of evolutionary framework by investigating new taxa in new environments. And I always wanted to be a marine biologist, so I basically looked for organisms that looked like these sort of interesting animal societies in the marine environment. And I ended up studying these fantastic fish, the clown anemonefish, Amphiprion, otherwise known as Nemo to many of you. So this is a beautiful photo. It's not my photo. It's taken by Matteo Visconti. But this shows you a classic clownfish group. These groups are small. They're anywhere from sort of 3 to 6 individuals. They're always found in close association with sea anemones. These are giant tropical sea anemones. These can be 0.5 meter to 1 meter across. They're not like the bitty little things off the coast here. Each group of clownfish is composed of a breeding pair and a number of non-breeders. And within each group, there's a size-based dominance hierarchy, which you can see very clearly in the photo here. So the female is always the largest. So if I had a pointer, I'd point to the sort of top left there. That would be the female. The one below her is the male. He's the second largest. And the non-breeders in these groups get progressively smaller. And clownfish are sequential hermaphrodites. They are first male hermaphrodites. So if the female of this group were to die, the male would change sex and become female. And breeding occurs year-round on a lunar cycle. And very conveniently for me, they lay their eggs next to the base of the anemone, so you can take photographs of those eggs. They're laid in a monolayer, and you can count those eggs, so you can get estimates of sort of reproductive success. Also, all the individuals in the population are very tied to their anemones, and they all differ in their color markings. You see they have these beautiful markings, the orange, the black, and the white. But everybody's so variable that I can tell them all apart. So you can track survival as well. So you can measure both survival and reproduction of these fishes, making them sort of great study organisms. And they're all at sort of scuba diving depth. Usually I work between sort of 1 to 20 meters. So what I want to tell you about today is a test of 4 different hypotheses: the kin selection hypothesis, future selection, ecological constraints, and social constraints. And then we'll wrap up with some conclusions, future directions if we have time. So let's talk about kin selection. So this hypothesis makes 2 critical predictions, and that is that the non-breeders enhance the fitness of the breeders. So yi is greater than yj, and non-breeders are closely related to the breeders. That means the coefficient of relatedness is greater than 0. And so to test prediction 1, I conducted a year-long study of 97 groups of Amphiprion in Medang Lagoon, PNG. This is my PhD work, ably assisted by undergrads from Cornell. And we monitored the survival, growth, and reproduction of 71 groups. And we conducted an experimental removal of non-breeders from 14 groups. And these experimental removal of non-breeders is sort of really important to get at this question, because often in these societies, the sort of quality of the dominance, the quality of the territory, and the group size is all positively correlated. And so you get a cause and effect, you have to sort of remove non-breeders and see what happens to the fitness of the breeders. And so this figure captures a lot of my PhD work. What you've got is control groups on the left and manipulated groups on the right. I did 4 months of tracked reproduction for 4 months before manipulating any of the groups. So that's before in the white. And then I tracked them again for another 6 lunar months after I'm doing the manipulation. And so, even though it's after, in the control groups, the non-breeders weren't removed. In the manipulated groups, the non-breeders were removed. And then on the y-axis, you have the eggs hatched per lunar month. And what you can see after years of painstaking work is that there is absolutely no effect of removing the non-breeders on the reproductive success of the breeders. And I think this is always a good moment in the PhD when you realize the really cool questions that you thought were going to be slam dunks turn out to be null results, and then everything becomes much more interesting. We also tested prediction too. So we developed 7 polymorphic microsatellite loci, cutting edge at the time, now basically obsolete, and genotyped 32 individuals from 9 groups. But that was enough to show us that the non-breeders are not closely related. Effectively, the coefficient of relatedness among individuals within groups is 0. So you can see relatedness on the x-axis, and frequency distribution, and it's centered smack on 0. And so there was another great null result from years of PhD work. So the take home is that in clownfish, non-breeders do not enhance the fitness of the breeders, and non-breeders are not closely related to the breeders, right? So perhaps as a little disappointing, but actually then things become more interesting. Because this means that kin selection doesn't explain the evolution of these non-breeding strategies in clownfish. And this is likely true for most coral reef fishes that form little societies like this, because most coral reef fishes have a dispersing larval phase that will break up kin association. Okay? And so non-breeding coral reef fishes must be contributing genetically to the next generation in some other way. And I think what's really interesting about this is, kin selection was sort of the primary hypothesis for the sort of what's driving the evolution of these societies in terrestrial systems. And it turns out not to be playing any role in marine systems. So you can then sort of ask, well, what are the limits on social evolution in the absence of kin selection? Okay, so let's talk about hypothesis 2, what I've called future selection or territory inheritance. And it basically makes one critical prediction. And the non-breeders will inherit their territory on the death of the breeder. So this FI term is greater than 0, right? It could be that your options out there are terrible and you're not breeding in the group, but if there's some probability of inheriting the territory, then you'll stick around. So to test this, I conducted a year-long study again, same year, 97 groups of clownfish in Madang Lagoon in New Guinea, and did a slightly different experiment where, first of all, I monitored naturally occurring breeding vacancies in 57 groups on 2 different reefs, but then I experimentally created breeding vacancies in 16 groups on 1 reef by removing the male, and then looked to see who took over that breeding vacancy. Okay, so this is one of the sort of simplest set of results I've ever published, but they're also super, super clear. So this is a table describing the pattern of territory inheritance. We've got natural vacancies and the experimental vacancies. And what you can see is that in no case did a non-resident, so a fish from elsewhere, usurp these vacancies when they occurred. It was always a resident non-breeder that filled the vacancy, and it was always the largest resident non-breeder that inherited the vacancy. Okay, and by the time I'd left the field, in all cases, these individuals who'd inherited this vacancy successfully bred. And so that showed that they inherited not just the territory position, but the breeding position as well. Okay. Yeah. I removed the male, and the reason for removing the male is I didn't want the results to be confounded by the time it took the male to change sex to become female. I just wanted to know, are the non-breeders inheriting a breeding position? Yeah. Thanks. Okay, so what can we say now? We can say that in clownfish, non-breeders stand to inherit the territory in the future. And so this sort of future selection helps to explain the evolution of non-breeding strategies in these clownfish. Future benefits are necessary, but they're not sufficient to explain the behavior of the non-breeders. The reason it's not sufficient becomes really clear when you understand how long-lived anemonefishes are. So anemonefishes, like naked mole rats, can live 20, 30, 40 years. And so individuals can be waiting 5, 10, 15, 20 years to inherit these breeding vacancies. Okay, they can be in the same anemone, twiddling their fins, just waiting for somebody to die in front of them so that they can get to breed. It would be a little bit like going to the supermarket checkout and waiting 20 years to check out. And nobody would do it. You'd either go to another checkout line or you'd do away with the people in front of you somehow. But these things don't happen in clownfish society. And so a complete understanding requires that we also explain why they don't disperse to breed elsewhere and why they don't contest for these breeding positions, given that they're waiting so long. So why don't they disperse elsewhere? Ecological constraints hypothesis makes two critical predictions. Individuals will stay as non-breeders when there is some ecological constraint such that the right-hand side of this inequality is close to zero, right? An ecological constraint can be things like, well, any clownfish, all the anemones on the reef in the tropics are occupied by other territorial clownfish, right? So there's no free habitat. It's also very risky to leave the anemone because it's providing protection from predators and move and look around and search for another anemone. Okay. But this hypothesis also makes the critical prediction that if you relax the ecological constraint by manipulating it, individuals should disperse. Okay, so in clownfish, two types of ecological constraint: the habitat is saturated and the movement between anemones is risky. Okay, so during my PhD work, we created habitat vacancies by removing non-breeders in that experiment that I told you about, and by removing males in that other experiment I told you about. But in these experiments, no individuals dispersed between anemones on the reef, even when these habitat vacancies were created. Right, so this is under sort of natural conditions. The anemones are evenly dispersed, not evenly dispersed. They're naturally distributed on the reef. They can be 100 meters apart, 30 meters apart. So essentially, the fish just don't have the information to know that there are vacancies out there, so they don't go out and search. Okay. Recently, a PhD student in my lab did a sort of finer scale test of this. She placed alternative anemones at 0.5 meters from focal anemones. And these alternative anemones had either just a male with which a non-breeder could leave to breed, or they were just empty, so the non-breeder could move straight into the female position in these empty anemones. Despite only being 0.5 meters away from the non-breeders, only 1 out of 32 moved. So even at these short distances, non-breeders aren't necessarily moving. And there's sort of a few potential reasons for this. One is they might just not have this in their behavioral repertoire, right? Which is a little bit like some social insects. They don't really go off to join other groups or explore other territories. So we wanted to get at some of these alternatives, so we flipped the experiment. And so here you see this, we're considering whether this non-breeder is going to move. And now what we did was we moved the non-breeder to the alternative anemone and looked to see if it would come back. And we did this at 0.5 meters and at 5 meters. And what you can see is that the non-breeder returned to its home anemone in 21 out of 32 cases. And this shows us that actually movement is in their behavioral repertoire. They will move if there's a reason to move. Although they didn't return home in any cases from 5 meters. And so this shows that as you ramp up that ecological constraint, as you make it more difficult to move, more dangerous, they're much less likely to move. And as I said, out on the reef, anemones tend to be 5, 10, 20, 30 meters apart. And so it's really very likely that under natural conditions, these non-breeding clownfish don't move because of this risk of movement, because of the ecological constraint. So what have we added? We can say that in the clownfish, individuals will stay as non-breeders because there are harsh ecological constraints. Okay? Individuals will disperse to breed elsewhere when this ecological constraint is relaxed. And so now we have sort of two things. We've got future benefits, explaining why they stay because they're ultimately going to inherit the territory. And we've got ecological constraints explaining why they don't leave to go elsewhere because it's dangerous. But we still haven't explained why individuals wait peacefully rather than contesting with the breeders. And so now we're going to talk about social constraints. And so this is an idea that's really sort of directly analogous to the ecological constraints idea. And the idea is that individuals will wait as non-breeders when there is some social constraint such that the right-hand side of the inequality is close to zero, such that the probability of winning a fight is close to zero. It also says that if you manipulate things, you should be able to manipulate things such that the likelihood of an individual contesting will increase if you disrupt that social constraint. So what might the social constraint be in clownfish? Well, one of the sort of interesting data sets that emerged from PhD, but that was never the main focus, but it's just what the data were, is that the subordinates in clownfish tend to be 80% of the size of their immediate dominant. You remember that photo I showed of the 3 clownfish at the beginning? Turns out that if you go onto the reef and you measure hundreds of clownfish, you can find that the rank 2 is always 80% of the size of the rank 1. The rank 3 is always 80% of the size of the rank 2. So this is shown in this figure here. You've got the size ratio between an individual and its immediate subordinate. And this is the frequency distribution. And so the purple here shows the naturally occurring distribution of size ratios. The yellow shows the distribution of size ratios that would be expected under a null model. And so what I did here is I took my population of clownfish, I randomized them on the computer, and I threw them back into groups randomly. And then I took the size ratios of those random groups. And this is the distribution of size ratios we would expect if individuals were randomly associated. So there's 2 big differences between these distributions. First off, you can see that there's an overabundance of size ratios around 0.8 in nature, and there's a lack of size ratios around 1.0. And what's actually going on is that subordinates are regulating their growth to remain 80% of the size of their immediate dominant, plus or minus 1 millimeter. They do it with millimetric precision. And this is, so I described all the sort of null results of my PhD that almost had me like throwing my computer out the window. But this was the sort of really exciting result, because this is a vertebrate, right, like you and me, that is turning its growth on and off in response to social context to be 80% of the size of its immediate dominant. Why do they do that? Well, we think they might do it because this is a plot of initial size ratio on the x-axis and the probability that the smaller fish wins on the y-axis. At 0.8, they have basically a 0 chance of winning a fight with their immediate dominant. And as they get bigger, so this is paired contests in the lab, that doesn't happen under natural conditions, their probability of winning a fight goes up. And so if you think about this from the dominant's perspective, the dominant is willing to tolerate the subordinate as long as the subordinate has no chance of winning a fight. Right? But dominants won't tolerate subordinates, they get bigger. And so to test this idea of social constraints, Rebecca, the PhD student, Rebecca Branconi, she took an individual out, or individuals out of naturally occurring size hierarchies, and she introduced individuals just a few millimeters bigger, which is effectively a relaxation of their social constraint, because now they have some chance of winning a fight. And a few millimeters smaller, which is basically strengthening the social constraint, because now they have even less. And what she showed is that when you relax the social constraint, the subordinate will actually contest for the breeding position. And so this is the weaker social constraint here, and the sort of 10 times out of 16, the subordinate contests. Whereas when the equal social constraint is stronger, that subordinate hardly ever contests. Okay. So the larger non-breeders contested, and they were evicted in 12 out of 16 cases, and the smaller non-breeders only contested in 3 out of 16 cases. So in the clownfish, we've got individuals waiting as non-breeders because there's this strong social constraint. But individuals will contest for breeding positions when the social constraint is relaxed. And so now we have the sort of complete picture, right? At least from the subordinate's perspective. You've got future benefits and ecological constraints explaining why subordinates will wait rather than disperse to breed elsewhere. And you've got future benefits and social constraints explaining why subordinates will wait rather than contesting for these breeding positions. Simply, they come in small, they settle into anemones with larvae, they grow to be 80% of the size of their dominant. They don't get any bigger because they would be evicted. And at 80% of the size of the dominant, they have no chance of winning a fight. So their only option is basically to wait for a decade. Yeah. So they, under natural conditions, they really don't contest because you have an established size hierarchy. A larva settles into an anemone. It will be allowed to settle into the anemone if the smallest rank resident is big enough that it doesn't perceive the larva as a threat. It then regulates its growth to only be 80% of the size of the one above it, because if it gets any bigger, the one above it just kills it. And so under natural conditions, what you see is effectively a very harmonious society, and it's only by tweaking the sizes experimentally that you see the fights elicited. And it's almost always the larger individual that wins the fight. It's just that as the size difference gets smaller, the probability of the smaller one winning goes up. Does that make sense? Okay, so we started off with an evolutionary puzzle, Darwin's one special difficulty, which appeared insuperable and fatal to his whole theory. But, you know, 150, 160 years on, we can say now as a field that 4 key concepts, really, kin selection, future selection, ecological constraints, and social constraints provide quite a complete understanding of the diversity of non-breeding and helping strategies observed in these complex animal societies. There is still outstanding questions for clownfish. One of which is, why do breeders tolerate non-breeders? Okay, from the non-breeder's perspective, there's nowhere else to go. You have to settle into an anemone and you have to wait, and that's it. So the breeders now have 1, 2, 3 non-breeders with them in this anemone. And you'll recall that I showed the non-breeders have no effect on the survival, growth, or reproduction of the breeders. So there's still this outstanding question of why the breeders tolerate the non-breeders. And this is a question that we're tackling now in my lab and with collaborators in the UK. So why do these breeders tolerate the non-breeders? They can evict them, they can kill them whenever they want, but they let them hang out. And so one way of thinking about this is just that kin selection and future selection might help explain why breeders might tolerate non-breeders rather than eviction them, right? So we're using the same sort of inequality, Hamilton's inequality again. We're thinking, what is it the breeders are getting out of these non-breeders such that it pays to tolerate rather than evict? So the first question people always ask us is, well, can the breeders evict the non-breeders? And yes, I've already shown you an experiment where we manipulate the size of the non-breeders and the breeders will evict the larger non-breeders. So if the non-breeders get too big, they evict them. We also did a similar experiment where we manipulated the reproductive state of the non-breeder. We took out rank 3 individuals and we brought in rank 3 individuals from other groups that were size matched, or males from other groups that were size matched. And we found that the males were evicted much more often than the non-breeders. So the breeders can detect both the size and the reproductive state of these subordinates, and they'll evict them if they try to breed or if they try to get too big. But they tolerate them most of the time, as long as they remain small and non-reproductive. So we can ask why. I've already showed you this figure. Non-breeders aren't helping in the present. They're not somehow providing what you'd see in birds or mammals, or sort of alloparental care. They're not helping tend the eggs or anything like that. So there's no gain in sort of the short-term reproductive success of the breeders. We know that non-breeders are not close relatives. So one hypothesis is that breeders might tolerate non-breeders if they are relatives who are going on to inherit their territory, right? We know they're not closely related, and for a long time we thought they probably weren't going to be related at all. But actually, as we've ramped up the number of genetic markers, and people have got better at this, we think there might be some subtle relatedness, such that the breeders are essentially tolerating great-great-grandchildren or third cousins who have come back to their anemone after being produced elsewhere. It's almost like some distant grandparent letting you inherit their house, right? So we think that might be fine. Another possibility is that non-breeders help in the future, and they can do this potentially in two ways. One is they could reduce the time for the breeder in the event that its partner dies. It could reduce the time to recommence breeding. And I tested that during my PhD work, and they're not very good as rapid mate replacements. Having a non-breeder around maybe knocks a month or two off the time it takes for a breeder to recommence breeding if it's widowed. But in the grand scheme of things, of the sort of 15, 20 years of breeding that this individual does, that 1 or 2 months is pretty trivial. What we think they might do, however, is not reduce the, not improve the mean reproductive success, but reduce the variance in reproductive success. And that's to say, if you don't have a non-breeder around, then and your partner dies, you may have to wait a very long time for a new recruit to turn up because of the sort of stochasticity of recruitment in the marine environment. Okay, it may be that somebody turns up very quickly, but it can be that you have to wait a year or 2 years for somebody to turn up if you don't have someone there in the anemone. And so what we think they're doing is basically keeping these non-breeders around as a bet hedging strategy to reduce the variance in the time taken to recommend breeding. Perhaps the most interesting idea, though, is that the non-breeders are helping in the future by nurturing the anemone. Okay, so the anemone is it's an animal that is providing protection to these clownfish, and it's quite possible that they're having non-breeders there enhances the fitness of the anemone, and that can happen in a number of ways. It's shown that the anemone fish produce nutrients that the anemone takes up. So nitrogen and phosphorus tend to be quite limiting, so the anemone takes these up. The anemone fish also defend the anemone against corallivores like butterfly fishes. They can also aerate the anemone at times of low oxygen on the reef. But just by swimming through the tentacles, they can break up the boundary layer and aerate the anemone. So there's all these potential benefits that the anemone might get from having multiple fish there. And as the anemone gets bigger and healthier, this feeds back to the breeders. So breeders tend to get bigger in larger anemones. Larger breeders tend to have larger clutches of eggs. And so there could be this sort of positive feedback where tolerating non-breeders enhances the quality of the anemone, and that improves the breeders' fitness. Okay, so with that, I just want to give some acknowledgements. So for permission and logistical support, the government of New Guinea, the landowners of Riwo Village, Christensen Research Institute, and John Mijau, my boat captain at the time. Also, more recently, I've been going to a different place in New Guinea, so the landowners of Tamare and Kilu, the Mahoney and Ndari Research Station, and Wilindi Plantation Resort, and our boat captain, Nelson Sikatua. Also, my PhD committee and collaborators, and a number of students and collaborators more recently. And with that, I'll take any questions. Thanks very much.
Just want to say that we're all available to help with your field work. Sounds
Okay,
good.
next year.
So why do the clownfish, so the clownfish can move back to the anemone they came from, but they don't move to a new anemone, even if it's right there and it's completely empty. Why is that?
Great question. So the question was, if I can go to the data, where is that data? So when we placed anemones just half a meter away, new anemones half a meter away from their regular anemone, they didn't move. So to see if they could move, we moved them to these neighboring anemones, and in at least half the cases, they moved back. 2/3 of the cases, they moved back. So it's a great question. Why are they moving back? We have sort of two ideas on that. One is it's pretty unnatural just for a large anemone just to crop up next to your own regular anemone empty. And this often happens after storms. And these anemones tend to be unstable because they're rolling across the reef. So from the fish's perspective, it could be that that is not a great alternative habitat. So they would rather be in their stable home. The other option is that, you know, they are in this anemone and they don't know how long it's going to take for other group members to arrive, and maybe having a large group is really important to sort of the success of the anemone. And so maybe it's just better to be in the larger group because that anemone is more likely to survive for a longer period of time than to be in a tiny group and you might not be able to defend an anemone. So we've got a couple of ideas that we haven't tested. It's a great question.
How is it that they're not related and yet socially and ecologically the constraints keep them from moving a lot?
Right. So they're not related because so the way it works is that their pair lays eggs next to the base of the anemone. The male cares for them for 7 days, then they hatch and they disperse for about 2 weeks, and they'll travel anywhere from 10 meters to 100 kilometers in that time. Okay, and then they settle back to anemones. But in the vast majority of cases, they're settling with their non-relative, right? If they're tolerated, as they settle in about 8 millimeters long into these giant sea anemones, they'll be tolerated if the largest non-resident is big enough that it doesn't perceive them as a threat. And if they're tolerated, they'll stay because it's really hard to find another anemone out there. And even if you did find another anemone, there'd be aggressive clownfish in it. And they wait peacefully rather than contesting because there's just no way for them to win that fight. So dominance through orders of magnitude bigger than that. For that, or did you have another element of it that you wanted to get?
They're moving a lot more after they've hatched.
Yeah, so they disperse as larvae. So that's when they're moving throughout the reef, and then once they've settled and metamorphosed and they're now sort of proper fish, then they never move again. No, that's it.
Thanks for a fun talk. I'm curious if there is something inherent about the marine environment that would change some of these social and relatedness dynamics. Like the species you mentioned that were terrestrial seem just as disparate as any one of those is from a clownfish. And the sort of argument you made at the beginning was these are all terrestrial environments. Let's look at it in a marine environment. What are the reasons that a marine environment could be different than a terrestrial environment?
That's a great question. Like, what are the primary differences? And I think, you know, the big one is this larval dispersal that's going on that's breaking up these kin associations, right? And so I think that's really fascinating just because it's this primary explanation for social evolution in terrestrial systems, because baby birds are born and then they have to make a significant effort to leave the nest, right? Or baby mammals are born and they have to make an effort to disperse, whereas baby fish hatch, they do disperse and they would have to make a significant effort to come back. So that's sort of the primary difference. But then I think there's lots of similarities, right? All these territory inheritance, ecological constraints, social constraints are exactly as we see them in terrestrial systems. And so I think that's really interesting to see what's different and what's similar.
That non-breeders do not settle with other conflicts that they're closely related to. So in what way would you test the close relation hypothesis that you had in your further discoveries segment? If like from what you have studied, they typically don't.
Yeah, it's a great question. So there's sort of an inconsistency in the talk where I've told you they're not closely related, and now I'm telling you that maybe there's a little bit of relatedness there. And the fact is, when when we first asked the question, we didn't expect them to be related, so we still did it with a minimal amount of data. Okay, so we used, I think, what did I say, 9 groups and 9 microsatellite markers, and showed that the mean relatedness is right around 0. It turns out that that's not enough microsatellite markers to detect really fine scale relatedness that you'd expect from like great-great-grandchildren or third cousins of around sort of 0.02 or 0.03. So basically, our early study showed they're not parents and offspring, they're not brothers and sisters. Our next study is asking, well, are they sort of second cousins and great-great-grand offspring? And we're using just more microsatellite markers and more data, basically, to get at the question of detecting at a finer scale. Does that make sense? Yeah, okay, great question. How do the fish know? That's the whole field in itself of sort of like kin recognition and things like this. In this case, they don't actually have to know. If just on average, occasionally you're tolerating a distant relative, then it can tip the inequality such that it pays you to tolerate everybody, even if you can't tell who they are. Just if one of those individuals in there is some distant relative, then it pays you to tolerate everybody because of the way the math is.
Hi. Do other reef fish use the hierarchical system that you've been using, or is that primitive for just this clownfish?
Yeah, so I have a really good collaborator, Marianne Wong in Australia. She works on emerald coral gobies, and they have a very similar system. And actually, it was really nice because when I worked on clownfish and sort of proposed this sort of crazy idea that they were turning their growth on and off, people didn't really believe it at the start because it's a bit far-fetched. But then she found exactly the same thing was going on in these emerald coral gobies, and they have a very similar social system, slightly larger social groups. There's also 27, 28 species of anemonefishes, all of which have sort of subtly different social systems. They have different anemone distributions in the habitat. They have different sort of life history traits, just like in, it's cooperative breeding birds, and then there's a genus of birds, and they all have slightly different social systems. So there's lots of scope there for people to do interesting additional projects that hasn't really been tapped yet.
Actually, a little bit related to what you just said. Do you have any understanding of the mechanism of how they regulate their growth and just turn it on and off?
I have, it turns out that this is much more common than we realized. So I was inspired by a paper that came out as I was writing on my PhD work that showed that marine iguanas can shrink in response to El Nino. Okay, so I was looking at this really funky growth data set, trying to make sense of it. And then I read this paper in Nature by Martin Wikelski for a tome. And essentially they showed that in El Nino years, male iguanas shrink skeletally, like their skeleton shrinks because they become nutrient limited, and they're trying to, and large males starve worse than small males, and so there's been really strong selection for this shrinkage. And so I thought, well, if iguanas could shrink, then my clownfish could certainly turn that growth on and off. And that's how it helped me make sense of it. But it turns out actually a lot of social vertebrates do this. So naked mole rats do this, meerkats do this, maybe even humans do this. And so the answer how, we don't know. I have a PhD student right now who's looking at sort of gene expression. We're doing little mini experiments in the lab where we get accelerated growth and experiments where we get decelerated growth, and looking at the genes that are turned on and off to try and get at this question of how they're doing. Yeah. Thanks. Got it.
Hi, so my question is just, like, why is it evolutionarily beneficial beneficial for the larvae to disperse immediately after hatching? Do you have any grasp on that at all, or?
It's a great question. I mean, there can be benefits just to dispersal per se, in the sense that you're avoiding competition with your kin. So if you're a parent and you put all your offspring in one place, then they're basically going to end up competing each other for the limited territories in a given area. So selection can favor individuals that disperse their offspring throughout the habitat. There can also be escape from parasites or diseases and things like this. These are other reasons that selection has favored dispersal. In the marine environment, it's also true that on coral reefs and other habitats, there's a lot of predators who hoover up larvae for breakfast. And so there's a hypothesis that it just pays to get those larvae out into the open water where they can feed and grow before they come back. It's a really hard hypothesis to test, actually, but that's the reason for their dispersal. And to think about what predictions that would make about what dispersal would look like. But it's one of the primary sort of dogmas in marine biology.
So you said that it's possible for like another female to come along, like if the original male would be widowed, and so then it might not like make the non-breeders necessary. But if they're not traveling after their larvae, then how would a new female be coming along?
Well, that was sort of the idea that let's think. When a female is widowed, so the male dies, it was possible before we did the experiment that you might see a non-breeder from nearby move, but it just turns out that never happened. Right? So they have two options basically. They either keep non-breeders there and then they can breed with that non-breeder as it comes through the hierarchy, or they can not have non-breeders and they can just wait for a larva to come in and grow up. It turns out other anemone fishes are interestingly different. So the pink anemone fish is very similar. It lives in the same sort of lagoons. It occupies the same tropical giant sea anemones. It has the same habitat saturation, but it's a much better swimmer. And there you'll occasionally see pink anemone fish move between groups. And that actually has really interesting consequences because it means that somebody's inheritance can be usurped, right? Like if I'm number 3 and the male dies, a bigger number 3 from next door could come over and usurp my inheritance. What's really seems to be going on is then there's less incentive to stay small in those groups, right? Because you don't want to stay too small, otherwise your inheritance gets usurped. And so they tend to get a bit bigger. And so those groups tend to have more conflict as well. So it's like a really interesting small shift in ecology results in sort of changes in these sort of internal dynamics. So pink anemone fish are just really nasty. I'll go on about pink anemone fish forever. Sometimes their groups get to be really big and there's more and more aggression and it looks like the female loses control of what's going on in the group. Okay, so she has all these subordinates who are just in conflict and she gives up. She swims down the reef to a beautiful clownfish colony and she kills everybody and she starts again in her own empty anemone. So just it turns out anemone fishes are really nasty. That's all I have to say about it.
Hi. Kind of on that note, I was thinking is you said the females are the largest, so they're kind of at the top of the hierarchy. Not vertebrates, but I was thinking ants and bee colonies, sometimes you'll get where they will kill the queens. And I was wondering, do you ever see that where they say like, you're old now, like move on in clownfish? And I guess secondly, when you're talking about contesting, is it the female that's kind of battling these contestants, or is it always the male breeding?
So two questions there. Do they stab the female in the back at any point? I've not seen that, and if it's occurring, it's a rare event. And so, you know, these rare events are really hard to detect. And probably the way to get at it would be to sort of do something experimental, like weaken the female somehow experimentally and see if there's some sort of contest of recognition. I mean, it's interesting because in a lot of social vertebrates, it's actually the female that's dominant. So this is true here. It's true in mole rats. It's true in meerkats. It's true in mongooses. So it's really interesting parallel between those, this system and the mammalian system. And then the other question was who is really doing the contesting, and it's usually individuals adjacent in rank. And so the time that I see evictions under sort of non-experimental conditions is always at the bottom of the size hierarchy, because everybody else has their sizes figured out. But at the bottom of the size hierarchy, you have the lowest ranked resident, who can be like 15 millimeters long, and then new larvae coming in who are like 8 millimeters long, and they come into conflict. And so you just see these tight fights between really, really tiny fish. While everybody else just doesn't care because they're like, go figure it out. But yeah, so those are sort of naturally occurring evictions, and then to really sort of see the evictions under at other times, you have to experimentally tweak the system. And it's really interesting. So that has led to a whole area called sort of hidden threats. And it's this idea that lots of sort of cooperative behaviors in animal societies are maintained by these hidden threats that you don't usually see unless you tweak the system. And we have very similar things in human society as well. And the example I always like to give is like the state trooper, right? Like if you're driving on highways in the United States, it makes no sense. They're long, straight, empty, and everybody's driving at 65 miles an hour, 70 miles an hour, not 90 or 100. Cars can do it, people can do it, nobody's doing it. Why not? It's because of this hidden threat that you're going to go past some bridge and you're going to get pulled over by a state trooper and fined for your lack of societal cooperation. So that's an example of a hidden threat in human society versus the evictions in clownfish society. This one? Yep.
I just like to know, like, based on like the social systems and climate change and other environmental stressors, like how's the population doing for the clownfish?
That's a great question. So what are the kind of climate change effects? In some ways, up until this point, been relatively lucky because these anemones are relatively unaffected by sort of marine heat waves relative to corals. And so while the corals have been decimated on some of the reefs I work on, the anemones are still intact. This year, there's been a massive bleaching event, but we have people in the field. And one of the things we're really interested in is, do clownfish buffer the anemones against bleaching, or do they help the anemones recover faster post-bleaching? Perhaps by doing things to help the anemone, providing it with nutrients, providing it with protection, and things like this. Yeah, I mean, this year in New Guinea where we work, the sea temperature's been like 5 degrees Celsius over normal, 10 degrees Fahrenheit over normal for a sustained period. So a lot I can say. I'm sort of watching the problem, documenting it, or collaborators watching it, documenting it. There's interesting questions we can ask, but there's not a lot we can do to prevent it from unfolding. Thanks.
Thank you all so much. Thanks for great questions. Don't forget, all of these talks are recorded. They're online. When you leave here, you can join us by joining these talks online and ask your questions online. So please do that. Next Tuesday, we have Fidel Adib from MIT. He's an associate professor there, and his lab group invents new wireless sensor technologies to interconnect, sense, and perceive the physical world in novel ways. And he has a whole subsection of his lab group that has an ocean focus. And he's looking at underwater GPS, underwater AI, underwater to air communications, and battery-free wireless underwater cameras. So some really cool ocean remote sensing talk next week. All of this is on our website, shoalsmarinelaboratory.org, and I hope to see you here or online. Thank you all.